Hello, everyone. Thank you for attending today's webinar, Circulating and Local Antibodies as Novel Cancer Biomarkers, presented by Cengenics. I'm Andrea Anderson, and I'll be moderating this webinar. I'd like to start off by introducing today's speaker, Jessica DeGama Duarte, PhD, Olivia Newton-John Cancer Research Institute. You can read her full bio on the left side of your window by selecting the Speakers tab. Just a few technical notes before we begin. You can access closed captions from the bottom right corner of the video player. This webinar is being recorded and will be available to watch on demand within 24 hours. We'd love to hear from you. During this presentation, please submit any questions you have using the Questions and Answers tab on the left side of your screen. Time permitting, we will conclude with a Q&A session. All right, let's begin. Jessica, please go ahead.
Hello, everyone. Thank you for that very kind introduction. It is an absolute pleasure to be speaking here today. I'll start off by giving a brief overview of what I'll be talking about. So I'll start off by giving an overview about what cancer biomarkers are and how we can use B cells and the antibodies that they produce as a form of identifying novel cancer biomarkers. And this might include diagnostic, prognostic, predictive, or even ones that are for disease monitoring and patient stratification. I'll tell you a bit about how you can measure these antibodies using a technology called protein microarrays. And then I'll talk you through a few applications where each application is a different publication, and it essentially covers a breadth of the different topics and different applications of the use of circulating antibodies. And then I'll very briefly touch upon some current and future work as well. So what are cancer biomarkers? The ideal cancer biomarker is one that is easily accessible, so a liquid biopsy, versus one that requires invasive access to tissue, and ideally one that can be used for either the diagnosis or the prognosis or to disease monitor patients as they either respond to treatment or progress or relapse. Similarly, biomarkers can be used to personalize patient stratification procedures to determine which treatment is suitable for which patient at which time. Now, in this schematic here, unfortunately, antibodies are not included as liquid biopsies, but if I had made this scheme, I would have definitely included them, and I'll try to explain why I think they are the perfect cancer biomarker. So B cells are a form, so they're part of the adaptive immune response, so similar to your T cells, and they also are part of the educated response that occurs in an antigen-specific manner. So when you have an antigen-specific T cell response, specifically a CD4 T cell response, you will also get a paired B cell response. When your B cells encounter a tumor antigen, they can develop into antibody-producing B cells, and they secrete antibodies against that cognate antigen. Hence, it's very valuable to be able to measure that output of antibodies that is circulating in the blood. Now, B cells are quite complex. There was a lot of contradictory data that came out initially, but hopefully now people are beginning to realize that B cells are indeed very valuable, just as T cells are. A lot of the confusion related to the use of pan-B cell markers, and that means that people would just use a pan marker such as CD19 or CD20 to identify all B cells. And of course, this does not distinguish between B cells that have pro-tumoral functions versus ones that have anti-tumoral functions. So it is very important to categorize your B cells very specifically based on the subset of interest and be able to distinguish between your effector B cells, which are your antibody-producing B cells, and for instance, your regulatory B cells that are making IL-10 and TGF-beta. So what can trigger the production of antibodies in cancer? This is quite interesting. So if you have just cell death in general, the release of dead cell debris into circulation itself can actually trigger an antibody immune response. Similarly, if you have a break in tolerance or a defect in inflammation, this too can also cause antibody production. But the more classic ones are altered protein structures or changes in expression level. So either a mutated protein, where the mutation is respective to the cancer, or a change in expression level, so overexpression of a tumor antigen. When the immune system sees these things occur, it triggers an antibody production specific to those antigens. Now, this is more complex than it seems. You can get different types of antibodies produced. Your high affinity antibodies will be IgG, but within IgG, you actually get IgG 1, 2, 3, and 4, where 1 and 3 are actually anti-tumoral, but unfortunately 4 and 2 are pro-tumoral. So at times, it's not sufficient to simply look at your total IgG production. It is important to also categorize these and look at different subclasses of IgG. Similarly, you can also look at an IgM response, which is your early immune response. So you always have an IgM response preceding a high affinity IgG response. So this also connects really nicely with tertiary lymphoid structures. Now, there is a lot of talk about tertiary lymphoid structures across the field. I am particularly interested in these structures because they are known to be antibody-producing sources, okay? So these structures mimic what occurs in the lymph node, but the structures form adjacent or within tumors. So that means that you do not have to go to the nearest lymph node in order to develop an anti-tumor immune response. This can occur locally and more rapidly. And the tertiary lymphoid structures are specific to that tumor type. So if you have multiple tumors across the body, a tertiary lymphoid structure in tumor 1 will be unique to that tumor. But importantly, B cells and antibodies are secreted into the bloodstream. So one of the main reasons why B cells are fantastic to be used as cancer biomarkers is because they give you a systemic representation of what's occurring throughout the entire body, not just in a single tumor. And this is quite important as well. Of course, antibodies can be produced locally as well within the tumor, and I'll touch upon that very briefly as well. So when we're talking about tertiary lymphoid structures, I just want to highlight that a lot of studies, similar to what occurred with B cells, unfortunately, miscategorize tertiary lymphoid structures. So they simply identify a B cell zone and a T cell zone and assume that that is actually sufficient to categorize them as mature tertiary lymphoid structures. But unfortunately, that is not the case. For a tertiary lymphoid structure to actively produce effector B and T cells and to release antibodies, it needs to be fully functional, have all the characteristics of a tertiary lymphoid structure, and also be mature. And we've developed a really nice protocol recently where we actually categorize these and give a series of markers that are important to adequately identify these structures in tumors. I'll show you how we've categorized these. So if you simply have an immune cell infiltrate without any aggregates, that's a grade 0, so that's definitely not a tertiary lymphoid structure. And if you start having immune cell aggregates, but no defined B cell or T cell zone, that's actually a grade 1 immune cell aggregate, but not a TLS. Now, the confusion often lies when you're looking at your grade 2 immature TLSs and your grade 3 mature TLSs. And mainly because these actually look quite similar, because you still have sometimes defined T cell zones, but for immature structures, these are often poorly defined. And importantly, there is no follicular dendritic cell network in the center of your B cells. So it's very important that tertiary lymphoid structures are adequately characterized, and often they go hand in hand with B cells with regards to being prognostic or predictive of treatment outcomes. And of course, we did a few staining across different human and also murine tumors, and we're able to identify both immature and mature tertiary lymphoid structures in both these cases. And I highly recommend this protocol, which is quite robust, and we've shared this across labs, and it works quite well, both in an automated and a manual fashion. And I've left the link at the bottom for that publication as well. But now coming back to antibodies. So why am I talking about tertiary lymphoid structures? So often, tertiary lymphoid structures have been defined as being the new best biomarker, both for prognosis and for predicting treatment outcomes, particularly with immunotherapeutics. But of course, what happens is that these are often confounded with B cell signatures and antibody signatures. Because, of course, tertiary lymphoid structures are these massive factories that are producing these antibody-producing B cells locally. One of the limitations of antibodies, though, is how you measure these. So, of course, you can measure them with ELISAs. They are less multiplexing, but they often can be used to screen circulating antibodies against different sets of antigens. However, protein microarrays are much more robust. So with protein microarrays, you can immobilize thousands and thousands of different tumor antigens onto usually a glass-coated slide. and use these to interrogate cognate antibody responses. Another reason why antibodies are such an attractive biomarker is because they are very stable in the blood. So this is quite important. So even if you're using samples that have been collected within 10 years, those antibodies are still stable and those samples can still be used after a long period of time, which is fantastic. Also, protein microarrays tend to use very minute amounts of serum or plasma, and I'll talk you through some of the applications that we've used. And in fact, we only use 1 to 10 microliters of plasma or serum in these experiments, which is very important when you think of how valuable patient samples are. So on this slide, I've shown a few examples of different protein microarrays that are commercial. And I'll just highlight the immunome protein array at the bottom, which includes 1,636 full-length human proteins. And these include proteins that are part of the immune response and tumor-specific as well. Now, one thing I haven't mentioned yet is that the arrays are actually fully functional. So even though we're using them to do screening of discontinuous epitopes and circulating antibodies in the blood, it is important to note that if I wanted to, I could actually do functional assays on these arrays as well. So how does the experiment work? So specifically with regards to the immunome array or another version from Syngenix, the cancer testis antigen array, these are printed onto a streptavidin-coated H slide using a robotic arm with different pins. And then a blood sample, in this case a diluted blood sample, is immobilized over the array. If there are any cognate antibodies to the immobilized tumor antigens, they will bind, and we can detect these with the fluorescent detection antibody. And here we can use IgG or IgG 1, 2, 3, 4, or IgM. You then obtain a fluorescent image, and you can quantify this fluorescence using a variety of different tools and software, including one that I've specifically developed myself, Protein Microarray Analyzer, to filter out any bad quality data and to correct for any batch effect and normalization so that you obtain unique profiles for each patient. The current gold standard for biomarker work is to determine the sensitivity and specificity of a specific biomarker. And ideally here, your sensitivity is the fraction of patients with cancer that a test can correctly identify as being positive if you're looking at the diagnostic biomarkers, whereas the specificity is the fraction of patients without cancer, so your healthy individuals, that the test correctly identifies as negative. So ideally, you want high sensitivity and high specificity when you identify your biomarkers of interest. I'll also just add in a plugin over here for blood cards. So another nifty tool that we have and can use is blood cards to immobilize a drop of blood onto them. And we can actually recover antibodies from that drop of blood from these cards. This is one example, but Syngenix does have their own as well. And this is very valuable because if you think of different rural areas, for instance, that can't access the clinic, you could actually just send one of these in the mail, collect blood, and ship it to your nearest lab without having to have patients actually go to the clinic themselves, which sometimes is a big limitation. So the two arrays that I'll be talking about are the CT100 array. So the CT100 array is a cancer-specific array that I developed during my PhD with Professor Jonathan Blackburn at the University of Cape Town. And the Syngenix cancer testis antigen array is very similar and an expanded version of this array. So it includes 123 cancer-specific targets. So that means when I screen healthy individuals, I don't get any hits. But when I screen patients with different cancers, I do in fact get different targets. The majority of these tumor antigens are cancer testis antigens, and the reason why these are so attractive is because their expression is not limited to a single cancer type. Instead, they are actually expressed across a variety of different cancers, which means that the utility of the tool can be applicable to different cancer types. And then the IOM array, or the previous immunome array from Syngenix, includes 1,636 targets. This number has probably been updated now. It tends to change often. And here, it is important that you do assay healthy individuals or in adequate control when you're doing your patient samples, just because, of course, some of these signals will light up in healthy individuals as well, because some antigens are autoantigens. They're not all just tumor-specific. So when you're looking at identifying diagnostic, prognostic, or predictive cancer biomarkers, You should do this usually with a cancer-specific array, just to avoid confusion. But sometimes if you need a more broad discovery-based approach where a larger array is better, then you should look at something like the IOM array. Also important when you're looking at anything like autoimmunity or immune-related toxicities that sometimes mimic autoimmunity. So one of the ways that people often tend to do is to use a discovery approach first, and then once they've identified their targets of interest, they can go onto a focused array where they just have different targets of interest for validation. Okay, so now I'll take you through a few different case studies. First off, starting with diagnostic and prognostic biomarkers and the identification of novel therapeutic targets. So this was a really interesting study that we did on cohort 1, which included 104 different melanoma patients that we had access to in-house at the Olivia Newton-John Cancer Research Institute. And here we identified roprin-1 and 1b as a novel immunogenic tumor antigen that could be used either as a therapeutic target or as a diagnostic biomarker for melanoma. And I'll try to convince you of this data as I talk you through it. So we have 4 different cohorts here. So we started off with our own in-house cell line cohort that includes 52 melanoma cell lines that we've established from patient tumors ourselves, for which we generated gene expression data. Our second cohort is the TCGA cohort melanoma that includes 469 different tumors and also has access to gene expression data. Our third cohort included tumor microarrays from 61 melanoma patients that we established in-house as well. And our fourth cohort included 104 melanoma patients with serum or plasma that we used to determine circulating antibodies. So here we've done an interesting approach where we've started with the gene expression data. We've confirmed whether in a large data set the gene expression data holds true. We've then confirmed if the matched proteins are also expressed, because at times you can have gene expression but no protein expression. And then we determined whether those proteins expressed were actually immunogenic, because if they're not immunogenic, you do not get any antibodies. So first off, we looked at our cell lines and looked at gene expression levels. And this is where we first saw this novel target, roprin-1 and 1b. And it was actually detected in 82% of cell lines for roprin-1 and in 84% of cell lines for roprin-1b. Now I compared this to CTAG1A, which is also known as NY-ESO-1 in a protein form, which was present in 95% of different cell lines, just because NY-ESO-1 or CTAG1A has been previously used as a cancer vaccine for melanoma. We then had a look at the TCGA dataset and confirmed the expression levels across an independent cohort, and actually confirmed that here roprin-1 and 1b was much more abundant. So 1 was present in 99% of different tumors at a transcript level, so virtually all melanomas, and 1b was present in 99%. And this was actually more frequent than CTAG1B, which was only really present in 68%, which is quite interesting. We had a look at whether there were differences in stages and even between genders, and in fact found that there was no distinction. So sometimes different CT antigens can be associated with a poor prognosis or the presence of metastases, but in this case that did not occur here. We went on to look at protein expression of our targets, and simultaneously we also added in just classic markers for melanoma, including mLANA, MelaNA, and SOX10, so they're often used as a melanoma tumor marker. And here we confirmed that the expression of roprin and ESO was predominantly cytoplasmic, which is important when you want to think of downstream approaches. And that was again using the third cohort of 61 patient tumors. We confirmed that we were able to detect roprin protein expression in 89% of patient samples that we tested, whereas NY-ESO-1 was only observed in 16% of samples, which was very interesting. So we are outperforming the current gold standard or the current gold tumor antigen in melanoma, which is ESO, by far. And one important thing was to determine whether the same cells were expressing ESO and roprin, and we actually found that often roprin 1b expression was detected ubiquitously, whereas ESO appeared more scattered and diffuse across tumor cores, and that will be important as well when you think of it as a novel therapeutic target. There were cases that had both roprin-1b and NY-ESO expression, but of course there were instances where you had exclusive expression of either. But if you consider including a cancer vaccine that includes two different antigens, so roprin-1b and ESO, you'll actually increase your coverage by 29% with regards to the specific cohort, which is interesting. So perhaps a multi-antigen vaccine is preferred over a single antigen vaccine. And now antibodies. So it's great that the protein is highly expressed, but is it immunogenic? Because if it is not immunogenic, then automatically that limits our use. So to explore the immunogenic potential of these new antigens, roprin-1a and 1b, we screened the serum or plasma from 104 melanoma patients using the CT100 cancer array. And here we found that roprin was actually detected in 71% of patients, which was higher than what we saw for NY-ESO-1 that was only detected in 64%. We saw co-reactivity in 52% of patients, but we did still have 17% of patients that didn't have reactivity for either antibody. But of course, when we included tyrosinase and melanase, which are the two gold standard protein antigens, we could then reduce this number down to 11%. So that means that we would have quite large coverage of our full cohort if we were to target an antigen vaccine, including those 4 different targets. And as mentioned, we looked at whether there was any differences between gender. We did have an issue with our cohort where we predominantly had stage 3 and 4 patients. So it might be that we do have a bias there, unfortunately, but we didn't see any gender-related differences nonetheless, or any stage-related differences. So that was a small study where we basically started from gene expression and then went through to antibodies. So once you've identified a novel gene target, if you want to see whether that gene target can be used as a biomarker, a circulating biomarker, you can start from gene expression data to protein expression data to antibody data, and that gives you a nice sequence or a nice representation of how to identify a novel immunogenic tumor antigen. And the uses are, of course, not limited to novel therapeutics, but also to diagnosis, because, of course, if the majority of patients do have these antibodies, these could be considered as a diagnostic tool. So that was looking at circulating blood, or so either plasma or serum. But of course, we defined a new method where we could actually look at tumor draining lymph nodes. And at the moment, we're also looking at tumors themselves. So from tumor draining lymph nodes, which the theory is that if that lymph node is draining that specific tumor, it essentially works like a tertiary lymphoid structure. If you isolate out that lymph node and culture it, you can actually get a source of purified antibodies directly from that lymph node that is produced into culture over 7 days. And you can use that source as a purified probe, we call it ASC probe, a purified source of antibodies that is unique to that tumor draining lymph node, and which might not necessarily match what you're detecting systemically, because systemically in the blood, you'll get a representation of all different tumors, whereas in this tumor draining lymph node, you will only get information on that specific tumor antigen makeup of that tumor where it is draining from. So using this method, we started off with a small study with cohort 2 in 20 breast cancer patients, and this was a collaboration with Cancer Probe. And here we were looking for novel tumor antigens, novel immunogenic tumor antigens. But in this case, we bypassed the gene expression and protein expression data and went straight to screening serum or plasma, as well as ASC probes for the presence of different tumor antigens of interest for breast cancer. And our cohort was rather small, but it did include a variety of different stages of disease in different breast cancer subtypes. And for the first time, we were able to identify tumor antigens that hadn't been previously identified in breast cancer. And we're also able to see that what was circulating in the blood was 10-fold lower in antibody titer than what we were seeing from the lymph node ASC probe. Also, we were able to see that every single antibody that was detected, so the specificity of the antigen in the tumor draining lymph node, was also present in the serum or plasma, but of course serum and plasma had additional targets that were not present in the ASC probe samples. So this was quite exciting, and it actually opened up our perspective a little bit, and we started looking at a different cancer type. So that brings us to cohort 3, 91 ovarian cancer patients, another collaboration also with Cancer Probe. And here we did something very similar, but we started with the discovery approach. So here we actually used the CDI Huprot array as well as the Syngenix CTA array to screen a cohort of 11 ovarian cancer patients. And here we had both serum plasma and also ASC probes from tumor-draining lymph nodes from these patients. And we identified different targets of interest. We then developed a custom array using CDI, where we only included 50 antigens that had been identified in the previous screens, and then looked at an additional cohort of 80 ovarian cancer patients, as well as healthy controls and benign controls, to see whether or not we could identify novel immunogenic tumor antigens or novel diagnostic tumor antigens for ovarian cancer. Here are the details of the cohort. So as mentioned, for cohort 1, we had the pelvic lymph nodes as well as serum, but for cohort 2, we only had serum. And similarly for the healthy cohort and the benign cohort, we also only had serum. So I don't want you to go through these very complex tables, but essentially the only important factor here is that, of course, we had many more hits when we were looking at the larger array, when we were looking at the focused array, but at the same time we did have some overlap between the ASC probe and the serum for that cohort, and we were able to identify targets of interest that were quite interesting that we decided to take forward into a custom array format. Now, this discovery approach sometimes does not yield important targets. So let's say that targets that are relevant for ovarian cancer weren't on either of the larger arrays. And there you'd have to take a step back and perhaps look at an even larger array, or look at some immunoprecipitation assays using ovarian tumor tissue samples where you can identify targets of interest that we might be missing. Because of course, with antibody profiling, you're limited to the content of the array itself. So this is probably the most important slide of that study. And here we were able to identify a series of different antigen specificities that really didn't discriminate between healthy individuals, those with benign disease and those with ovarian cancer. We were able to see some that were present in benign conditions and ovarian cancer patients, but not in healthy individuals. And this is quite interesting because if we can identify some markers of benign disease, or probably these are markers for early cancer changes that are at the time still deemed as benign, but have probably already turned to a cancerous state from an immune system perspective, this might also be valuable, particularly because benign conditions can often be confused with cancer. But importantly, we identified a series of different targets that were unique to ovarian cancer patients and absent in benign individuals and those that were healthy. But unfortunately, we were only able to identify 29% of the cohort of 80 ovarian cancer patients. We had a higher sensitivity for mucinous and serous subtypes, but of course, less than 30% isn't an ideal diagnostic biomarker. But I will say that currently for ovarian cancer, there is none. So I guess 30% is better than 0%. But of course, this isn't attractive enough to warrant a subsequent study or to guarantee additional funding, unfortunately. So now I'm going to shift gears a little bit, looking at predictive biomarkers of therapeutic outcomes. So markers that can distinguish patients that are responding to treatment from those that will not respond at day 0, so before treatment has even started, as well as a means of monitoring responses to treatment on treatment. And why is this important? So a lot of people in the immunotherapy setting talk about hot and cold tumors. And I really love that this paper back in 2016 by Yuan et al. Actually listed antibody responses as one of the key factors to distinguish between an immunologically unresponsive or cold tumor from an immunologically responsive one or hot tumor. So a hot tumor is known to have a lot of immune cell infiltrates, but also a robust general antibody response. And to sweeten it even further, they actually specify protein microarrays here as the monitoring strategy, which is fantastic. So the ideal scenario will be to separate responders from non-responders before treatment has started, but even so, to look at whether patients would benefit from a single agent or a combination agent of each immune checkpoint blockade marker. And the main immune cell checkpoints that are relevant here are PD-1 inhibitors and CTLA-4 inhibitors, or a combination of both. And these work directly on T cells, but of course can affect B cells as well, even though not much is known about how they affect B cells specifically. That also brings me to immune-related adverse events. So immune-related adverse events occur as a result of immunotherapy, and they actually mimic autoimmunity. So they look quite similar to autoimmunity clinically, but at times there can be drastic differences as well. So essentially you tend to get that cytokine storm, an abundance of antibodies being produced into circulation, at times in a non-specific manner. And these can occur across the body, so from the skin to the brain, to the lungs, to the gut. And there were a few interesting studies that actually identified that you can see B cell changes between patients that had these toxicities when compared to patients that did not. And that this could separate the probability of survival from these toxicities really nicely, and even perhaps identify specific toxicities or organs that would be affected based on the targets. So that brought us to a cohort 4, which was a small study with 5 advanced melanoma patients. And this study was actually discontinued because of the side effects. Patients received intralesional BCG followed by CTLA-4 blockade. So essentially, tumors were injected with BCG, and then patients were given an immune checkpoint blockade agent, so immunotherapeutic. 2 out of the 5 patients actually had severe high-grade toxicities, including colitis and hepatitis, and the trial had to be discontinued. And so we first approached Syngenix with this cohort and said, look, what if we could use the immunome or IOM arrays to predict these toxicities? If in fact we think this is mimicking autoimmunity, can we see an abundance of antibodies being produced for these patients? And very interestingly, just from the starting point, we had different time points. So during the BCG induction stage, as well as the CTLA-4 inhibitor stage. And you can see both in the table and in the plot, in red we have the patients that had these toxicities, and in black we have the patients that did not. And this is quite interesting because you can see that you have, for the 2 patients, patient 4 and 5, that had these toxicities, one actually had no antibody specificities light up before treatment, but these were induced very strongly at day 49, and then lowered slightly. Whereas for another patient, we actually had pre-existing abundant antibodies. Now, at times, patients can have undiagnosed autoimmunity. That might have been the case here. It's difficult to say. But when that does occur, these patients will, without doubt, nearly always develop these toxicities. And the different antigens that were lighting up here were not just autoantigens, they're also CT antigens, which is quite interesting. So autoantigens and tumor antigens. When we looked at the timeline, and we used proportional pie charts here, so the larger the pie chart, the more antigen specificities are lining up for these patients. You can see that for patient 1, 2, and 3 in A, there was really no change in the amount of antibodies or the antigen specificities that were detected. All these patients had progressive disease, none benefited from treatment, unfortunately. But in B, you can see for patient 4 and 5 that either they started off with an abundant number of antibodies or this then developed. But importantly, what I want you to see here is that the increase in antibodies or antigen specificities preceded the clinical detection of these toxicities, which is very important. So from screening antibodies, you can actually see that you can actually predict which patients would have these immune-related adverse events well before this adverse event is clinically apparent. And that has a lot of utility in the clinic because at times these toxicities can actually be fatal or lead to long-term consequences in these patients. In addition to having to discontinue the treatment, at times the complications are severe and they have to live with them for the rest of their lives. When we did some clustering analysis on these, we could very easily separate patient 4 and 5 from the other patients as well. So we were quite confident that we could easily distinguish both at baseline and on treatment, between patients that had these toxicities from patients that did not. Now, of course, this was a collaboration with Syngenix, and we have subsequently engaged in additional studies looking at the immunotherapy setting across 2 different cancer types. I can't share that data just yet, but hopefully soon. The data does look equally as exciting, both with regards to predicting treatment outcomes and also regards to predicting toxicities. And finally, the last cohort, the second last cohort, I think, cohort 5, looks at 46 melanoma patients that were actually treated with an ESO vaccine. So remember in the first cohort I mentioned NY-ESO-1. So here we actually used the array, in this case the CT100 array, to look for evidence of immune responses to a vaccine. So you can see exactly just how versatile the use of antibodies are across these different settings. So patients were given an ESO vaccine in melanoma, and we used the antibodies to screen patient blood to look for on-treatment induction of immune responses, both against the target antigen that we were injecting, but also across other antigens. And in fact, we saw that ESO antibody titers increased following vaccination with an ESO vaccine, as well as CTAG2, which is a highly homologous tumor antigen. And importantly, we could see evidence of determinant spreading across different antigens post-vaccination. So this is actually quite valuable if you want to see if a vaccine is having the desired effect of inducing immune responses. And this would work for other immunogenic antigen-based or peptide-based vaccines as well. And now is the last cohort, so cohort 6. And this was a cohort of 45 metastatic colorectal cancer patients that were treated with dual angiogenesis agents. And this was a clinical trial here at the Olivia Newton-John Cancer Research Institute. And here, again, we wanted to do something similar, so measure whether there was an immune response to the treatment in these patients, but also see whether or not we could predict which patients would benefit from treatment. And interestingly, we could see here that there was, in fact, a global increase in all antibody titers observed after 6 weeks of treatment, which is quite interesting, and that this compared really nicely to baseline. And this was after the treatment, which included, in this case, VEGF-A and Ang2 inhibitors. Similarly, we saw evidence of determinant spreading, as we saw with the NY-ESO vaccine as well, which is very interesting. So not only are we triggering an immune response with this treatment, but that is spreading across different tumor antigens, which is more valuable. And interestingly, when we did some clustering analysis, there was one patient that was an exceptional long-term responder. And interestingly, he separated really nicely from all the other patients, which meant that we could actually predict this patient as being different to all the others and as having a good outcome. And this was probably one of the first indications that we received that we could predict treatment outcomes with some agents, and not just your classic immunotherapeutic agents, but also anti-angiogenic agents. Similarly, we could actually also predict patients that progressed rapidly at 3 months versus those that did not. And only one patient really didn't follow this line. And of course, with the clinical trial, you're limited in the patient samples and the patient numbers that you have. So in this case, of course, the cohort is small. So ideally, we'd like to look at a larger cohort to validate some of these findings. And with that, I'll just highlight some of the current and future work that we're looking at. So as mentioned, we're looking at 2 different cancer types undergoing immunotherapy with checkpoint inhibitors, so specifically melanoma and red cancer cohorts. And some of the key questions that we're asking, both with Syngenix and also with the University of Cape Town, is looking at whether there are differences in the antibody repertoire between pre- and post-immunotherapy treated samples, whether we can, of course, identify responders from non-responders at baseline, so before treatment has started, and whether we can predict immune-related adverse events, which is quite important as well. Similarly, we're looking a lot at the early stage setting, so early stage melanoma, prostate cancer, and ovarian cancer cohorts. We also now have a much larger breast cancer cohort that we just received funding for, and we're screening different antibody titers in early stage patient samples to see whether or not we can use the arrays or different tumor antigens as diagnostic biomarkers. Importantly, we're quite interested to see whether or not antibody titers disappear when you have curative surgery, Or if when you have residual disease, these levels remain there, but at low levels. And of course, because protein arrays are much more sensitive than your classic ELISA, we could potentially see them. And similarly, whether they increase or not with disease recurrence. Another side of this is, of course, the antibody-independent B-cell tumor immunity. So I mentioned tertiary lymphoid structures. It is important to note that if you are identifying tertiary lymphoid structures as being predictive or prognostic, you're probably overlooking that there is a big B-cell and antibody component there amongst your data. But similarly, it is important to categorize the role of B-cells in the tumor microenvironment. Antibody-producing B-cells are just one subset. There are many others that are just as important, including the regulatory subsets. And of course, it is important to look at both the circulating antibody repertoire, but also the local antibody repertoire, and not just IgG, but also IgG1 and 4, IgA, and IgM. Similarly, of course, tertiary lymphoid structures are still very important, and we're doing quite a lot of work, particularly in lung cancer at the moment, looking at whether these structures are predictive or prognostic. And we've also done this in in prostate cancer too. And with that, I will just acknowledge my lab, the Tumor Immunology Lab at the Olivia Newton-John Cancer Research Institute, particularly the lab head, Andreas Barron, as well as the research assistant that assisted with a lot of this work, Luke Quigley, as well as the clinicians and funders. So we received a lot of funding across different agencies, including Melanoma Research Victoria, Australian Prostate Cancer Bioresource, Victoria Cancer Biobank, Twitter Cure, Earlier.org, the CAS Foundation, Cure Cancer, Cancer Australia, and the Lung Foundation Australia. And of course, we work very closely with Syngenix, particularly with Jonathan Blackburn and Katie Leonard. And I've added my contact details at the bottom, both my email address and my Twitter handle, so please feel free to contact me if you have any questions. I'm happy to answer them.
All right, thank you for the presentation. Let's move on to the Q&A now. As a reminder, please submit your questions using the Q&A tab to the left of your screen. We have a lot of great questions already, and we will try to get to as many as possible. For the first question, how reproducible are the arrays? What cancer types can be investigated using the arrays?
Yeah, that's a great question. We've actually done some studies where we compared a few arrays side by side to look at how reproducible they are. I'll limit my response to the Syngenix arrays and the CT100 arrays and say that they are extremely reproducible. So for each array run that we do, we always include a positive and negative control, and we tend to use the same sample repeatedly, and that array profile is identical every single time. Now, I will say that that doesn't always occur across other protein microarrays that are commercially available. And in fact, we've had experience where data is not reproducible at all. So I can very confidently say that these arrays are highly reproducible, which is a definite plus and exactly what you want. With regards to the cancer types, we have looked at a lot of different cancer types so far. As I mentioned, the cancer testis antigens are expressed across a variety of different ones. The reality is that so far, I haven't found a cancer type that doesn't have antibodies against at least one cancer testis antigen. The sky's the limit. But of course, this is all restricted to solid tumors.
Another question, why consider IgG subtypes and IgA, IgM profiling local and circulating antibodies?
Yeah, that's a very interesting question. So unfortunately, at the beginning, everyone kind of just looked at total IgGs. You miss a lot of information, unfortunately, particularly when you're looking at prognostic biomarkers. So identifying patients that are high risk or low risk, or even identifying patients that will respond to treatment. Imagine if you have amongst your IgG an imbalance between your IgG1 and 4 and mainly a pro-tumoral phenotype rather than an anti-tumoral phenotype. Similar to your pro-tumoral immune cells and your anti-tumor immune cells, it is important to know which IgG subtypes are being produced because that will affect the effect that they have on the tumor. So that means if you mainly have stimulatory IgGs, that's probably good, and those IgGs are aiding in the anti-tumor immune response. But if you mainly have suppressive ones, then they're doing bad. So it's not important to just look at your total IgGs. It is also important to look at the different subclasses. And why look at local and circulating? Well, I guess that's an interesting one. So it is important to know how the tumor microenvironment is different from circulation. Now, my go-to will always be serum or plasma because it is a systemic representation of all tumors for that patient. But at times, if you want to look at just how heterogeneous the different tumors are, or if there, for instance, is a tumor that is progressing, whereas others are responding well to treatment, then it is particularly important to know exactly what's happening at that progressing tumor that isn't responding to treatment. And you can do this really nicely by just looking at the local immune repertoire of those tumors and get key information about which antigens are causing harm instead of doing good.
Another question, can circulating tumor-specific antibodies be used to detect residual disease or disease recurrence?
Yeah, I'm currently trying to answer that exact question in a cohort of breast cancer patients. I've done a bit of work in melanoma as well. The initial hypothesis was that if you have detectable antibodies after surgery that was meant to be curative, that means that there is residual disease somewhere and that the surgery was in fact not curative. However, we've actually found across two different cohorts now that once there is an immune response there, your memory B cell response continues to produce antibodies. So that means that even if you have cleared the tumor, you will still get a low level production of antibodies against the different tumor antigens that have been cleared. Now, of course, that means that you probably are limited in looking at residual disease here. But you can look at disease recurrence because when disease comes back, you get spikes in these antibody titers. The higher the tumor burden, the higher the antibody titer. So you can actually track really nicely when disease progresses or recurs. But yeah, not too sure about the residual disease setting just yet. I'll need to generate a bit more data in that respect.
All right, another question. Why are circulating antibodies predictive of immunotherapy outcomes or toxicities?
Yeah, that's a really interesting question as well. So, I guess when initially we looked at immunotherapy, the general consensus was that if patients have an active immune response or an active immune system, if your immune system has seen the tumor and recognized it and triggered an immune response against it before treatment starts, intuitively, that means that patients are likely to benefit from immunotherapy that will further enhance that pre-existing immune response. So essentially, if you have circulating antibodies before treatment has started, that means that you're looking at a patient that probably has a hot tumor with a lot of immune cell infiltrates. But of course, if your immune system has not seen the tumor or triggered any immune response or antibodies for that patient before treatment, giving a patient immunotherapy will not boost any pre-existing immune response because there is none. And in that case, patients are better off doing, let's say, chemotherapy or radiotherapy before they actually do immunotherapy to try to trigger that tumor to becoming a hot tumor so that the tumor is seen by the immune system. And only once you then have an antibody response or a T cell response, then you can boost that with immunotherapy agent. So essentially, we're just getting a representation as to whether patients have that active pre-existing immune response or not. A snapshot into whether the immune system has recognized that there is a tumor there and is actively trying to fight it. And then with regards to toxicities, I think here it's essentially patients that have too much of an immune response. So an overactive immune response are the patients that actually tend to have these immune-related adverse events. Whether this might be because they have undetected autoimmunity or something else that has happened in their prior treatments, because often patients have been heavily treated before they get immunotherapy, depending on the different cancer type. It's intuitive as well. So if patients have a lot of these antibodies before they started, so an excessive amount, you can nearly guarantee that these patients will have toxicities. And so whether patients are given a reduced dose or accompanied by steroids or closely monitored, whatever it might be, then that's an important information that the clinician needs to have in order to treat these patients to avoid these toxicities that can be life-threatening.
All right, well, that was our final question. We had a lot of great questions today and couldn't get to them all, but we will try our best to get back to everyone who submitted personally after the webinar. Thank you for attending this Fierce Biotech webinar and submitting so many great questions. I'd like to thank our speaker for participating and Sengenix for presenting today's webinar. A recorded version of this webinar will be available for you to access within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.